You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Metzger. He's the Chief Scientific Officer of Tierra Biosciences, recently renamed from Synvitrobio. Maybe possibly the rename was that this name's a lot easier to say and better. But Louis, thanks for coming. How you doing?
Very well. Thanks for having me, Rich. Synvitrobio was a difficult name because people always mixed up the syllables. And it would be
like
vitro synbio and so on. So we've rebranded as Tierra, and for a number of different reasons that I can discuss later. But we thought it was an easier to remember and sort of a cleaner name.
Yeah, no, it makes sense. Yeah. Well, tell me about Tierra. What's the premise of the company?
So historically, many chemicals and enzymes that are used for various biotechnology purposes, whether it be, insecticides in agriculture tech, ag tech, or even, drug-like molecules that are natural products that are used in biopharma. They've often been produced by expressing them in host organisms, or more specifically, expressing enzymes that catalyze the production of useful or interesting chemical products. And they've been done in cells. So yeast, E. coli are common host organisms, some types of plant cells, isolated human cells, and so forth. But one limitation of that sort of bioproduction, and especially the prospecting for new biochemistry, is that when you make a compound in an E. coli or yeast cell, or the enzymes that catalyze the production of that compound, and those enzymes or that compound are toxic to the host cell, it dies, or it finds a way not to produce the enzymes that you want it to produce, and thus you don't find what you're looking for. And so cells really regulate themselves quite well because they've evolved to do that. And it puts a limitation on what types of proteins one can express and what type of chemicals one can find in host systems that we're used to working with, living organisms that produce what we want them to. And so Tierra Biosciences is really founded on the basis of grinding up cells, preserving the biochemistry that those cells do, specifically those cells' ability to take DNA and make a message from it, which is RNA, and then take that RNA and produce proteins. Proteins can be products of their own that are used for specific purposes, or they can be proteins that are also enzymes. So enzymes are proteins that catalyze chemistry. We find that our thesis is that we can express proteins in these cell-free systems that are derived from living cells, but are not in fact living. And we can uncover proteins and products of enzymatic biosynthetic pathways that would normally be toxic to the host. So what we're really doing is we're trying to uncover this chemical dark matter, as it were, that is encoded by the genomes of all the organisms in nature, but that humans haven't been able to easily produce or access in host cells because of toxicity to the cell. So in a nutshell, that's what we're aiming to do. And that's what we think and have some evidence is a unique role for this cell-free expression technology.
Well, I was reading some of the notes. Yeah, this would be a funny example, but if you had a whole bunch of cells, could you process them and extract like the ribosomes and make like ribosome juice or something, or mitochondria juice, where you had, you extracted just the mitochondria from, several million cells and put them all together and see what you saw, or, the ribosomes. And are you able to isolate, for instance, specific organelles from a population of cells or other features intact?
Yes, we definitely are. And the process for doing that, for isolating organelles, is nothing that we invented. It's been done for many years for different types of organelles and different types of cells. And we've found isolation of specific organelles quite useful. And for instance, one can isolate bacterial ribosomes in case those are limiting in, say, a bacterial cell-free system. And for eukaryotic cell types that we're interested in, we certainly can isolate quantities of subcellular organelles that have useful properties in these cell-free systems. And one can imagine what those might be, especially from an energy regeneration standpoint.
I have a weird question. Have you ever tried to isolate parts of a cell and see if they, quote unquote, live, if we can sustain them, feed them, and keep them going? Has anyone tried to do that?
Well, in essence, that is what we do. It's a bit different if you compare that process for, say, bacteria versus eukaryotic cells. Given the higher level of complexity of eukaryotic cells. But if you start with the bacterial lysate and think about what it's doing, it has everything it needs in the cytosol to do transcription of DNA and then translation of DNA with the ribosomes, and then chaperones to help the folding of proteins and the various cofactors that you need to power the biosynthetic pathways to do all of this. So we basically preserve the ability to convert DNA to protein. Whether that's really life, it's self-sustaining but not self-replicating. And I think that maybe that's the fine difference. And there's no homeostatic feedback. So that is to say, the cells can't repress, or the cell lysates can't repress the expression of something that's toxic to them because, by breaking them open, we removed, much, but certainly not all of the biochemistry. And particularly, we preserved, this transcription and translational biochemistry that we really want. And in eukaryotic cells, it's much the same, although what that practically looks like is much more dependent upon what system we're working with. And even among the bacteria that we've studied and made cell-free lysates out of, and others that have been reported in the literature, one sees a wide variety of behavior, but at its essence, these systems work well when they can perform transcription and translation. And sometimes you can separate those two steps. Sometimes you can do them all in one pot. But it's almost alive, but not quite, I guess, is how I think about it.
So what are some of the things you're trying to elucidate by, by, I guess, isolating certain parts of, bacterial cells?
So we're really trying to make things that a bacterium or a eukaryotic cell would not want to make if it was fully self-regulating, if it was alive. So one category of these things that we're really interested in and that we've had, I think, a significant amount of success with are enzymes that do useful biochemistry. So biochemistry that would be helpful if one was going to use green chemistry to do some industrial process to make some valuable molecule, or one step in the synthesis maybe of that molecule might be best catalyzed by an enzyme that you find in nature. The problem is, if you're going to test those enzymes, you have to, clone them, expression clone them in a way that you can put them into your host organism. So say it's an E. coli or a living E. coli or a yeast, and then make that enzyme and break open the E. coli or yeast or other type of cell and analyze that lysate for whether it contains an enzyme that you tried to express, and if that enzyme does what it's supposed to. Our system is much, much faster and able to cover more ground because the cells are already broken open. We just use lysates, but they can allow us to quickly put in genes in expression cassettes and ask, can we make a gene that might have an industrial use, a gene product that might have an industrial use, and can we make many variants of that? So we can print these open reactions and ask very quickly, from this panel of genes that putatively encode an enzyme that does something useful, which of those variants of that enzyme express, and then we can test, or our clients can test those produced enzymes for the ability to catalyze some very particular and useful chemistry. And the key point, I think, is that we've been able to express enzymes that are really toxic to most host cells. And with these cell-free systems, we can express them. They catalyze the chemistry that one would expect them to. And this is a really powerful discovery tool for these types of enzymes. So that's one application. Another application that we're excited about is that if you think about how you could quickly test different panels of enzymes, ability to catalyze, for instance, a small molecule that might be of biotherapeutic usefulness, certain types of natural products, for instance, that nature via enzymes can catalyze the formation of very easily, but humans working in a fume hood have much more difficulty making. And so these are often molecules with handedness and shape. So we think that our pathway our platform is really well set up to querying whether these biosynthetic clusters make what we're interested in, because since these are open systems, the molecular biology is much easier than, say, expressing a series of genes all together in a living cell. So we can take linear constructs of single genes and pile them all into a single reaction, and do various combinations of them, and find out in what conditions and with what orthologs of some of the enzymes we can make an interesting natural product. So there's sort of two branches here. There's this functional genomics branch for discovery of enzymes that are hard to express in living cells. And on the other hand, there's this branch of small molecule discovery and sort of rapid prototyping of biosynthetic pathways. And because these are open systems, we can also put in unnatural substrates. So small molecules that would normally be hard to get into a cell, and thus have enzymes made in that cell, acting upon those substrates, we can feed those into our tiny reactions, and they're open systems. And so we can make unnatural natural products in this manner. And so I think it opens up the opportunity for the ability to, cover interesting corners of chemical space.
So some of the things that make it difficult to do this with the whole cell is what homeostatic drive of the cell, which I guess includes keeping toxicity at a minimum. The properties of a molecule coming through the membrane or not coming through, and migrating to the right spots, and maybe
exactly.
I don't know if there are any, but defense mechanisms of the cell itself. Maybe there's internal ones, certainly external. I guess there's a lot of factors that make this another viable way to look at reactions.
Precisely, and I think it's a useful. The platform is a useful tool to add to the many tools that the whole field of biological engineering or synthetic biology, whichever one wants to call it, has built. So I think that it is an additional tool that is complementary to the really beautiful work being done by companies such as Zymergen and Ginkgo and Amyris and Hexagon Bio, Lodo Therapeutics, and others to really start prospecting in essentially the genome of the world of like all sequenced organisms for useful biosynthetic pathways that allow us to make chemicals in a way that humans in the fume hood can't make. And I think we're going to discover many, many useful things, using these sorts of platforms. And the cell-free platform enables a certain type of discovery that in-cell platforms don't. Cellular platforms, of course, are much more scalable immediately if one wants to say, okay, we have a cell that produces something that we want because we've expression cloned a whole pathway into it. One can then grow up and that cell on in huge quantities and extract the product. And that's what Zymergen and Ginkgo and other companies like that are doing. And what Tierra Biosciences is doing is we're saying, look, we're not really at this point in a large cell-free scale-up production system. We have this discovery platform, so we can help you find orthologs of enzymes really quickly that you might not be able to find otherwise, and then we can take those high-value findings and then engineer those more if we need to scale them up. And at the same time, we're taking a longer view of can we use this process to really discover libraries of new chemical matter that would be useful for agriculture tech or biotherapeutic screening. And with something I discovered when I worked at Novartis Pharma in its infectious diseases division is that 2 or 3 million compounds in small molecule libraries in pharma are maybe not as diverse as one would want them to be. So I think that there's a place for discovering natural product inspired small molecules, ones that are enriched in chiral centers and ones that are often made enzymatically by nature. And so I think our platform fits into sort of the discovery of enzymes and the discovery of diverse chemical matter in the spectrum of what's going on in biological engineering.
Right. So have you seen any companies that have made like a ribosome farm or a mitochondria farm? They've taken, tens of thousands of these organelles and put them in like one gigantic cytosol bath and had them operate, kept them alive, and had them, churn out molecules or process molecules?
Well, it probably has been done and is certainly doable. I think a lot of that sort of work often falls into the category of different companies' trade secrets. But I can point, one can point to a number of companies in the cell-free space that must do something of the sort in order to scale their cell-free production of interesting molecules. And a good example would be Sutro Therapeutics, and they do large-scale in vitro production of antibodies. And for a number of reasons, that may be an advantageous way to make them as opposed to cell culture. So I would guess that Sutro probably has a very robust energy regeneration system and probably the use of organelles. And I think everyone who's working in the cell-free space is thinking about, what are ways that one could cleverly, fractionate cell lysates and do clever supplementation of those lysates and even mix and match them. So there can be components of one cell-free system that might be good, for instance, for folding your protein of interest, or maybe installing post-translational modifications. That's chemistry that's put onto a protein that's in addition to just the amino acids that are normally incorporated. And so there's this combinatorial, large set of combinatorial possibilities that makes the field really exciting because it's an open system, so we can supplement it as we wish. Extra ribosomes, we can do that. Organelles from a different cell type than the one that we're primarily using to drive our cell-free system, that can sometimes be doable. So I think there's much to do and much to learn in this space.
Interesting. Well, you have any? I don't want you to give any trade secrets or anything, obviously. But are there any specific examples of molecules you've been able to process or create using your system, or are you developing more just the platform of how to do it, and it's for other companies to do specific applications of it?
So we have a number of internal projects that will probably be proof of concept projects for our Series A fundraising round. And I can't really talk about those. However, the company for some years now has done a number of contracts and grant work for federal agencies and for NGOs and so forth. And I think one example that I can give that's not public yet, but we will be publicly reporting it because we're required to as a condition of the grant, is that we're doing some work with the Bill and Melinda Gates Foundation in the enzymatic synthesis of a natural product that's used in the treatment of malaria. And in particular, there's one enzyme, there's a cytochrome P450 that is key to catalyzing the production of this molecule, but it has never been reported to be expressed in, for instance, E. coli. And one reason that we found out that is because it's so toxic to E. coli that the E. coli put in stop codons in the open reading frame. This protein is so toxic that you can't even expression clone it. But we've been able to make variants of this protein, and it's a plant protein, in some of our different cell-free systems, and show that it can be produced at high levels, and it has the expected activity. And we think this is pretty exciting because it's an enzyme that I think people have wanted to work with historically and have not been able to, probably because of its toxicity to many hosts. But the other thing that's neat about it is, as a cytochrome P450, it has a heme group that has to be installed. And the lysate in which we're producing the cytochrome P450 preserves the ability to synthesize heme and install it in the cytochrome P450 enzyme that requires it essentially as a post-translational modification. So this is really exciting. And in fact, when we express this protein, which ends up having a reddish color, as many cytochrome P450s do, one can watch the cell-free reaction go from sort of a clear or light yellow color, which most of our systems are, to increasingly pinkish orange as it makes more and more of the cytochrome P450. So it's so we can spectrophotometrically follow the production of that one anyway. And so that's the sort of thing that really excites us. I can also mention, though I can't discuss the particulars of the project, that we recently did a multi-phase project for a large chemical tech / ag tech company, a Fortune 50 company. And they gave us a panel of very difficult to express proteins. And these were of interest to them for a number of downstream applications. And we were able to express many of this panel. And unbeknownst to us, a number of other companies were asked to express the similar collection of this, difficult to express proteins. And ours was by far and away the most productive system. So we had the most proteins that had the correct function in the analysis of our client. So we're really excited about that. And it gives us confidence that we can uncover things that are difficult to express in cells by doing them with these cell-free systems. So that was more of a functional genomics application, but the details of that I'm obliged to keep under wraps.
Right.
But it's exciting.
I've heard of organ on a chip systems and even interlinking certain organs on chips. What about an organelle on a chip system? Has anyone conceived of that, where you take certain cellular functions and try to, recreate them on a chip and maybe interlink one or two of them or three of them and see if you can use that as a proxy for generating, or at least seeing what happens and why certain compounds can't be made by the cell or can be made by the cell?
So this is a really good question. And again, these sorts of things have been done by a number of groups in academia and industry historically, maybe not explicitly thinking of recreating the entirety of a cellular function on a chip, but more from the perspective of building a really complex biochemical assay on a chip, which inevitably involves the assembly of protein, complex protein complexes to do specific biochemistry for screening purposes. And one sort of example of this use that comes to mind is there's been a number of good works over the years on producing protein arrays on chips. And so, if, for instance, you wanted to produce every variant of, say, a human drug target for which there were many variants, and the biochemical differences in these variants affected how that drug target, say, bound a drug-like molecule that was known to bind to it and affect its function. One way to do this is print DNA on a chip and then use in vitro transcription translation reactions to produce the proteins that DNA encodes all in one well, and then wash away, conjugate that protein to the chip surface in some way or another. And there's many options for doing so. Wash away the in vitro, the cell-free transcription translation reaction, and now you have your protein or protein complex variant on a chip. And so certainly, one could elaborate upon that even further and make very complex, proteinaceous local environments, in a chip conjugated way. And certainly that's another use of our technology that we're pursuing, because I mentioned that we have a number of internal proof of concept projects. Some of those are in the area of biotherapeutic discovery, or at least identification of classes of chemical matter that can be made enzymatically but are hard to make in the fume hood. And if we're going to make panels of small molecules, it's also helpful to make panels of targets against which to test those small molecules binding, or if they're enzymes inhibition. And so in vitro transcription translation reactions are really ideal for making targets on a chip. And so you put your finger on another application that we've piloted and we're really excited about, because it's directly useful to us in our proof of concept studies. Longer term, I think it could be directly useful to especially the biotherapeutic discovery world, because one of the difficult parts of an early discovery pipeline in any sort of therapeutic discovery is the in vitro treatment of the target. So if you're screening a library of small molecules, or if you're panning for an antibody against a particular target, you need to have a version of that target or versions of that target that behave well in your assay or are amenable to downstream characterization. And traditionally, proteins have been expressed, and I did quite a bit of work in protein expression when I was at Novartis, making these drug targets in a way that they were well behaved. But again, you have to expression clone them in living cells, grow up those cells, slice them, isolate and characterize your protein target of interest. We think that we can cover a lot more ground testing many, many constructs of valuable targets using cell-free transcription translation. And again, this has been done historically, in some quarters, but it's perhaps not as mainstream as one would imagine. And so the technology's there; it just needs to be applied. And I think we'll see more and more of that in the future.
Okay. Any surprises in doing this processing of cells? Have you looked at, for instance, the prevalence of certain compounds that are supposed to be within, a particular type of bacterial cell, and seen any outliers, or have you seen any compounds that you didn't know were there?
Well, we've certainly seen the preservation of pathways that we weren't sure, biosynthetic pathways native to the host cell that we weren't sure were still working after our treatment of the cells, and conversion of them from living cells into these, transcriptionally and translationally active cell-free lysates. And one example that I mentioned earlier was in a given system, the preservation of the robust ability to make and install heme into cytochrome P450s. That was a surprise in the system in which we tested this. I think we've done some metabolomics work, so looking at small molecule metabolites and how they change over time in these cell-free reactions, and that's guided us. And we've been surprised, I think, by the determinants of the kinetics of these reactions. Some things were surprises, some things when we thought carefully about them and basic biochemistry made quite a bit of sense. I will say that it seems like a simple system and can be described as a simple system, any cell-free system, but actually they're very complex. And every variable is important for making them reproducible. And so I think that one thing that we found surprising and really required a great deal of engineering, but in retrospect is not so surprising, is just how carefully we have to control the exact volumes of components that we add to these cell-free reactions, and and the methods that we use for doing that really have had to be carefully tested and calibrated so that we get consistent results. And so I think chasing consistency in cell-free transcription and translation is not as straightforward as it would seem a priori, because you no longer have self-regulating cells, and so everything from reaction volume to, not surprisingly, oxygenation or lack thereof, to numerous other variables have to be accounted for. And so we've spent a great deal of time engineering consistency in our cell-free transcription and translation work. And it's not a finished process, but it's getting more and more robust with less variance from run to run that we do when we express test proteins. So I'd say the engineering aspect and the automation and the data handling aspects have also been maybe not surprises, but definitely something that have required as much investment as we had suspected, and probably now even more.
Okay. Well, very good. What do you expect may happen? What are some milestones for the company and for you over the next year or two?
So we have a number of different milestones in different categories. So we have a functional genomics arm or cluster of projects in the functional genomics space, and this is the discovery of useful enzymes. And we have some contracts with large chemical tech and ag tech companies for which we've provided deliverables, but we're continuing on to negotiate next phases of those contracts. And of course, on others, we're waiting back to hear how our system worked in their hands, and did the enzymes that we produced, were they functional or interesting? And so I think as we get more and more of those data, we'll have an interesting case to make about our platform as a valuable functional genomics platform. And I think that might look like continued and expanded partnerships with the people with whom we're already working, and and probably some other collaborations that are in discussion now. At the same time, our purely in-house set of proof of concept projects are ongoing, and we'd like to have a very clear readout on one of our key small molecule discovery proof of concept projects this autumn. And so we'd like to end 2019 with a clear proof of concept or two built around using our system to discover difficult to synthesize natural products, small molecules on one hand. On the other hand, have shown traction in the functional genomics space, helping to turbocharge collaborators and customers' functional genomics hunting, as it were. And so those are the two things that we'd like to see come to a point by the end of the year where we can, early next year, say, look, we have these proof of concepts now. What do we want to get into big joint research agreements with partners? Do we want to raise a Series A based on internal data? We'll see what the data and what the timing suggests we should do. So it's nice to have a number of different options. And in addition to that, we have a number of grants and contracts with government and non-government agencies, and those provide revenue, and then our delivery on those helps us build the case for moving more and more towards building our own molecules in-house, our own molecular libraries, and partnering with private sector collaborators.
Okay, very good. And what's the best way for folks to get in touch or to ask questions?
So they're welcome to visit our website, tierrabiosciences.com. Certainly, they're welcome to reach out to Zach Sun, the CEO and founder of the company. He's reachable through the website and on LinkedIn. And certainly, listeners are welcome to contact me via our website, via LinkedIn. My email is Louis, Louis, at tierrabiosciences.com, and certainly welcome inquiries and questions.
Okay, very good. Well, Louis, thank you for coming on the podcast. I appreciate it.
Yeah, thank you, Rich. Really appreciate it. Nice talking with you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.